Dr. Fish's credentials are almost as awe-inspiring as her accomplishments. This is an incredible lady, and I've had the pleasure of hearing her speak on more than one occasion. Dr. Fish is a professor of immunology at the University of Toronto. She's an emeritus scientist at Toronto General Hospital. She received a Bachelor of Science from the University of Manchester, a Master's of Philosophy from the University of London, and a PhD from the Institute of Medical Sciences at the University of Toronto. Wow. She has been at the forefront in the battle against AIDS, Ebola, SARS, arthritis, and now COVID-19. And she has received many international awards and has been published and quoted on cytokines, the immune response, and the use of interferons in numerous scientific journals. So Dr. Fish is going to tell us today whether this is the new normal, and she's going to talk about how students play a role in changing our course. So I'm going to turn it over to you. Welcome, Dr. Fish.
Thank you. So it's my pleasure to participate in this, and thank you all for showing an interest. And as Harris has indicated, what I'm hoping to do in the next little while is walk you through how it's possible to take a career from being a student to going into a wet lab to do research in an academic environment, and how you can translate your wet lab research into real life clinical utility. And then at the end of my presentation, what I'd like to do is throw out there suggestions in terms of, if you have a scientific background, whether it's an undergraduate, a graduate, a postdoc, or whatever it is, what you can do with that scientific background, obviously related to science and beyond. So with that, I'm going to now go ahead and share my screen and start my presentation with all of you. And hopefully you can see this now. I just need a yes from somebody, please, Julius or Harris.
Yes, we can.
Okay, excellent. So let me begin. So I'm going to walk you through from bench to bedside. Now, what I want to do here is I want to minimize, okay, I can do that way. Okay, so from bench to bedside, talking about how this really is a journey from being a student all the way through to somebody as long in the tooth as myself, and tell you about how you acquire those skills, and those skills really have utility, widespread utility in the real world. Okay. So what I show you here is a map, a global map, just to remind us that although we're going through this SARS coronavirus 2, this COVID-19 pandemic right now, we really should be aware that viruses have caused outbreaks, epidemics, pandemics for as long as we've been recording them. And in this world map, what I've shown you is a number of those viruses. So you're all familiar with HIV, where there are some 40 million people around the globe have been infected. But what I'm going to focus on today is very specifically SARS-related viruses. And you can see, again, from this particular animation that I've put up, that when SARS coronavirus emerged in 2002, there was a number of fatalities around the globe, some 774. Certainly in Canada, we were one of the countries that were affected. And then in 2012, MERS, that's the Middle Eastern respiratory syndrome, another coronavirus emerged. And after that, at the end of that particular outbreak, and there are still some cases, there were 858 deaths globally. For neither the SARS or MERS was there a vaccine or an antiviral. And now here we are, we find ourselves with SARS coronavirus 2 or COVID-19. Now, this is a far more serious pandemic, and I don't have to tell you that there are now over 3 million people around the globe infected and hundreds of thousands have died. So let's just reflect a little bit before I go into the science and my journey on just to remind everybody about these coronaviruses. So on the left-hand side of this panel, you can see a number of common respiratory coronaviruses which circulate seasonally and give us just a really mild cold, not even flu-like, but a mild cold virus infection. And all of these coronaviruses originated, to the best of our knowledge, from bats. Now let's look at, on the right-hand side in red, I've highlighted this MERS coronavirus, SARS coronavirus, and now SARS coronavirus 2. Again, originating from bats. What we know is with SARS coronavirus in 2002, bats likely, which are asymptomatic, infected these civet cat-like animals, and then those animals, the virus jumped from them, jumped species to infect humans. And this is actually, SARS coronavirus is far more fatal than this new coronavirus, I just want to point out. In the MERS infection, we went from bats to camels to humans. And now we find ourselves in this ongoing pandemic with SARS coronavirus 2. So let me tell you about how my journey started. So I'm an immunologist, so I'm aware of the fact that for any and all virus infections, the very important immune response is our innate immune response. So my career has started with cell biology and an understanding of immunology. That's the theoretical background. And what we've learned is in the first 24 hours, the first days after you become infected, it's absolutely critical. And that innate immune response, that early immune response, determines the outcome. And my research over the years, unfortunately over decades, has revealed that this immune response, this critical immune response, is absolutely dependent on what we call a type 1 interferon response, interferon alphas and interferon beta. So I spent many years working in the lab trying to understand what that interferon response is. And what this slide, for those of you who are familiar with cell biology, what this slide, this cartoon shows is that viruses, as you know, can infect any cell in our body. We can have respiratory viruses like flu, coronaviruses. We can have viruses that infect the heart, cardiotropic, viruses like hepatitis that infect the liver. Pretty much any organ can be infected. You have viruses that enter through the skin through mosquito bites. So we have to have some sort of a surveillance system in place that will allow us to detect when a virus enters into our body. And what we've learned is that there are many different recognition systems, but what they all do, they're called pattern recognition receptors. What they all do is recognize that foreign pattern, that genome of a virus or a bacterium, and we're going to focus on a virus. So it doesn't matter what the cell is, and it doesn't matter where that organ is, and it doesn't matter what the virus is, but what this cartoon shows you, that all those TLRs, dectins, nods, MDA5, doesn't matter where they are on the outside of the cell or in the cell, once they're activated by that genomic material, what we see is that it triggers an incredible signaling cascade. And what I want to point out to you is that the end point of that signaling cascade is the induction of these type 1 interferons. It doesn't matter what the virus is, doesn't matter what the cell is, but the end point is once those pattern recognition receptors are activated, it triggers an interferon production. And I spent many years figuring out where those receptors are, which viruses trigger which of those receptors. But the point is, what we learned is that interferon is turned on. And why is interferon turned on? It's because And I don't know why it's not turning on the animation here. Okay, what we learned was that interferon binding to, once interferon is produced, then what does it do? Well, it's secreted by those cells that are infected, and it touches all the neighboring cells, some of which are infected and some of which not, and it touches them to trigger a very specific receptor called the interferon receptor, and that's in the top of that cartoon on the very outside of that cell. And again, you can see that once those yellow circles, those interferons bind to that receptor, again, there's this incredible network of signaling. So that signaling results in a variety of modifications. One of the first things that happen is that those signals trigger in the nucleus what we call histone modification to open up various DNAs such that you can get transcriptional activation, or genes that are turned on that are responsive to interferon treatment and that are absolutely critical for an antiviral response. So these genes, the ones that we know of in the right-hand bottom corner, we know those genes, once they're turned on, produce products that are important for an antiviral response. And again, I spent many years trying to figure out, as a research scientist early on in my career, how that receptor was activated, what those signals that were turned on as a result of that receptor activation, and again, what the genes were involved in. So initially, it was a lot of cell biology that I focused on. So subsequent to that, we've learned that not only are genes turned on, but in order to respond to a virus which replicates very fast, you have to turn on what we call mRNA translation. You have to make those proteins that will be involved in killing the virus. So interferons trigger a whole number of signaling cascades, and the end point is that those cells that are activated by interferon have what we call an antiviral state. And in this slide, again, it's a cartoon, and all I really want you to take away from this, that in addition to interferons being directly antiviral, in other words, they'll prevent viruses from entering cells, they may degrade the viral genetic material by chewing up the RNA or DNA. Again, these are the gene products that are turned on. They will prevent viruses from uncoating. So every virus that may have an envelope, in order for it to replicate in a cell, it has to uncoat that envelope to expose the genetic material. Well, there are interferon-inducible gene products that block that. Can't uncoat, can't replicate. So there are multiple stages where interferons are directly antiviral, and that depends on the virus. At the same time, interferons turn on an immune response. And I don't want to belabor this, but again, this took a lot of research in my lab and in other labs. We did all the direct antiviral studies, and then we asked the question, do interferon alphas and beta, do they turn on an immune response? And what our data show that yes, they absolutely do. So they, for example, will stimulate hematopoietic stem cells. So those the very earliest cells of an immune response, before even those immune cells have been differentiated to do their task. They'll turn on those stem cells to proliferate, and interferons very specifically activate different immune cell populations. Natural killer cells, as their name suggests, are involved in killing. Macrophages, which are involved in perhaps ingesting and degrading viruses. Dendritic cells, which are really important because they take various components of the virus and present it to other immune cells to invoke an immune response. They activate B cells to become antibody-producing cells. And we've heard a lot about the excitement around producing vaccines or using plasma that will generate antibodies to kill the SARS coronavirus 2. They activate T cells also, which are important in clearing a virus infection. So again, what my research has done in my lab, working with graduate students that I've had and postdocs and research associates, We first figured out all the signaling. Then we asked the question, how do interferons actually, from their gene product, block virus replication? And then went on to show that not only do they directly inhibit virus replication, but they also activate a number of immune cells to clear virus. So that's why, in response to any and all viruses, we turn on an interferon response, because it is so pleiotropic. It has the capacity to interfere with virus infections at multiple sites. So now let's focus on the work that I then went on to do during the SARS outbreak. So knowing that we had here what we call a broad spectrum antiviral, all our research has brought us to that stage, all our in vitro and preclinical, that's the small animal models, we then asked the question, okay, let's look at this particular virus and figure out what's going on. So in 2002, 2003, at the end of the outbreak, we had over 8,000 people around the globe who'd been infected, and some, as I mentioned, 774 deaths. On the right-hand side, I show you the numbers in Canada. So this virus traveled on an airplane. It was an index case from Hong Kong who came to Canada, and that's how the virus spread, respiratory virus again. 251 cases across Canada, mostly Vancouver and Toronto, and 43 deaths. Notably, at the time of this outbreak, there was no vaccine and no antiviral drug. Okay, so the time it takes to make a vaccine, as we're learning now, even if you move as fast as you can, it's about 12 months. So there's that window when nobody is protected, and what we're doing now, we're self-isolating, we're using personal hygiene, but the best case scenario would be to have an antiviral that we could use. But during the SARS outbreak, we had neither. Now, this particular virus is this, in common with all viruses, encodes in its genome some factors which very specifically block interferon production. I listed them here, NSP1, NSP3, ORF6, and M. I defy any of you who are listening to this webinar to identify a virus that does not have in its genome factors that will block interferon production, which tells us all the more how important interferon is for clearing virus infections. So armed with this knowledge, we went ahead and as we do as research scientists, we first did some preliminary studies in the laboratory where we used an isolate of a patient who was infected with this particular virus, and we challenged cells, human lung cells, with this virus. And then we asked the question, if you use increasing doses of ribavirin, which was the standard of care for patients at that time, or increasing doses of an interferon, which I've listed here as interferon alpha-con1, what is the effect on inhibition of SARS coronavirus? And that's on the y-axis. And I think it's fairly convincing that if you look at the red plot, ribavirin does not kill or inhibit replication of the virus at all, whereas interferon alpha, as I would anticipate, based on all the knowledge that we've gathered over the years, is very effective in inhibiting the virus. So now you have to take the leap of faith and saying all the in vitro data we've done, all the preclinical data, does this allow us to do a clinical study? So you translate your research into clinical utility, and that's exactly what we did in Toronto. So what I'm showing you here is some X-rays of the lungs of patients who were infected with this SARS in 2002 in a hospital. They were hospitalized in Toronto, and the upper panel of 4, what you can see in those X-rays, you see that white area, which gets cloudier and cloudier from day 9 to day 18. It's effectively the lungs become more and more occluded, become blocked. It's a respiratory infection, right? And the more blocked they become, the harder it is to breathe, and that's why you need oxygen supplementation and even intubation. In the lower 2 series, these were patients who received interferon, daily interferon, and you can see how over from day 9 or day 10, their lungs progressively improved. So what this plot shows you, from the worst chest X-ray abnormalities to 50% resolution, you could see in the black plot that interferon resolved we got to that 50% in 4 days versus the control group that didn't receive interferon in 11.5 days. So that was interesting. At the same time, what we learned, and again, this is using now what I had to switch gears from my basic science knowledge. I had to start acquiring some clinical expertise. What we learned was that there was a particular blood biochemistry marker called creatine kinase, which absolutely spiked in patients who had severe respiratory distress with SARS. Those are the open circles on the left-hand panel. And yet every patient who received interferon, those are the closed circles, black circles, they never had that spike. And indeed, if you use, this is a respiratory disease, so you want to think about oxygen saturation in the blood. If their oxygen is well saturated, that means their lung capacity is good. We saw that for all the patients who received interferon, they had good oxygen saturation. Those are the closed circles again on the right-hand panel. And for all of those who didn't receive interferon, their oxygen saturation levels fell. Many of them required nasal prongs to oxygen for supplementation or intubation. So these data showed us we could take our in vitro and preclinical data, our evidence that we had, and translate it into clinical utility with this particular SARS virus. So now let's switch gears. And again, here I am at a stage in my career where I've got a lot of information about interferons. They're widely available. They're being used for hepatitis C, but not for acute virus infections. We had some data on SARS. We also did some work with Ebola where we showed it was effective, but now we're with SARS coronavirus 2. So as of a few days ago, as I mentioned, there were over 3 million cases around the globe. Well, close to, I think it's now close to 220,000 deaths. We know the cells that the virus infects, and we know that compared with SARS, where there was an approximate 10% fatality, we estimate, based on testing for confirmed cases, that the fatality rate is actually lower with this particular virus. And what's intriguing, and this is a recent publication, what's intriguing is that this particular coronavirus is far more effective at multiplying than SARS coronavirus. So can you see that upper plot? The orange is SARS coronavirus 2 and the blue is SARS coronavirus. And what you can see is that the multiplicity of infection, it multiplies much faster over 48 hours than SARS coronavirus. And what's also intriguing, remember I mentioned to you that all viruses inhibit interferon production? Well, this particular coronavirus 2 is even more effective at doing that than SARS coronavirus 1. And that's what the 3 bottom plots show you, that if you look at the blue triangles, there's a modest induction caused by the virus of interferon alpha and interferon beta. But if you look at SARS coronavirus 2, you can see that pretty much all of interferon alpha and all of interferon beta doesn't, you don't get that kind of induction. So this virus is very effective at blocking interferon production. So what does that inform us? Again, we have a broad spectrum antiviral interferons, which should be able to override this inhibitory effect because you come in and you treat with interferon. So what I'm showing you here is a chest X-ray of a 68-year-old woman admitted to a Toronto hospital with fever and worsening shortness of breath. And she was confirmed positive by a PCR swab 3 days prior. And what you can see by the yellow arrows, remember I showed you that chest X-ray series for coronavirus 1? Well, here again, we see that the chest X-rays, there's notable peripheral airspace consolidation. Again, this white opacity in the upper and lower lobes. So again, this is a severe respiratory infection and this is how patients present in hospital. So again, you're talking to a basic scientist here who is now learning throughout her career some clinical stuff so she can translate. So I've been able to translate my basic science research to clinical utility. So what did this prompt us to do? So it prompted us to actually, at the start of the outbreak, it prompted me and my group to contact some colleagues in Wuhan in China and to undertake an exploratory study. Now, what you have to realize is during a pandemic or during an outbreak, and certainly at the very start of an outbreak, it's extremely difficult to set up a randomized controlled trial, which is the gold standard for approval of any drug, because remember, there was no vaccine or no approved antiviral, and there still aren't. So what we did was an exploratory study, because the question I asked was, will interferon be effective as it was with SARS coronavirus 1 against this SARS coronavirus? So I contacted some colleagues in Wuhan, specifically in one of the hospitals, and we agreed to undertake an exploratory study and look at the effectiveness of interferon treatment in patients in hospital with confirmed COVID-19. So between January 11th and February 12th, we evaluated 77 patients. Some of them received an aerosol, an inhaled interferon. Some of them received the standard of care at that time throughout China, which was a drug called Arbidol, which we've subsequently learned is completely ineffective. And others received a combination of interferon plus this Arbidol. Every patient, we took a throat swab because you need to confirm that they're PCR positive and when they clear the virus. So you take serial throat swabs. We took CT scans. Again, this is a way of looking at their chest, their lungs, measure their temperature, oxygen saturation, and a range of other blood biochemistry, cell counts, and cytokines. So once again, we're taking a drug where we've done some basic science, fundamental science discovery, all the way through from preclinical work into a clinical scenario. And what I want to mention is that none of the patients that were recruited in this study exhibited signs or symptoms of what we called end organ dysfunction. So none of them were in acute liver failure, acute kidney failure, and no patient, regardless of their treatment, developed kind of respiratory distress that required them to be in an ICU or intubated. So I'm just going to run through the results and let you know that actually these data were submitted for publication, because as scientists, the best way to disseminate evidence is to submit to scientific journals for publication, where it will be peer reviewed by 2 to 3 reviewers, and there'll be this iterative process of them passing comments, asking for new experiments, asking for new statistical parameters, other ways to present your data. And only once a manuscript has undergone peer review and been accepted, is it really appropriate to stand up and say, these are our data, this is the evidence we have, and share it widely with the scientific community, and obviously also with the broader community. So what these data show is the time essentially to viral clearance. And there are different plots. So on the left-hand side, the blue plot is the time it took for patients to clear their virus when they received Arbidol only. And the red and green plots are for Arbidol plus interferon or interferon only. What I want to reiterate is that there are publications that have emerged which have suggested that Arbidol treatment is ineffective. So although we didn't have any controls that received nothing or placebo, perhaps it's reasonable to suggest that the blue plot is actually those that, because the drug was ineffective, their natural course of disease. And on the right-hand side, what we did was we combined both groups that received interferon as the red plot compared to the blue plot. And the key here is it's a significant difference. And in fact, it took on average 7 days less to clear virus for all those patients who received interferon. And if we adjusted for age, for sex, for comorbidities, the data remained absolutely significant, p-value of 0.002. We also looked at a couple of parameters which are associated with the severe inflammation in the lungs. And what this slide serves to illustrate, again, blue is Arbidol only, and red is those patients who received interferon, either alone or in combination with Arbidol. And what these slides show you is that there is a significant reduction in circulating levels of IL-6, which are absolutely associated with acute respiratory syndrome, and a reduction in what we call C-reactive protein, another measure of inflammation. So not only did interferon treatment reduce or accelerate clearance of virus from the upper respiratory tract, but it also reduced the inflammatory response. And this was with daily inhaled interferon, okay, twice a day. And again, the data remained significant even when we adjusted for those patients that had comorbidities, male versus female, and older versus younger. So in summary, we used although there are limitations, this was a small cohort, it wasn't randomized, treatment arms were of unequal size. We showed that interferon treatment accelerated viral clearance, it reduced IL-6 and CRP levels associated with inflammation, and these findings are now the basis, this exploratory study, for a randomized control trial. So we've gone from basic fundamental science research, preclinical, clinical, and now we're moving globally into randomized control trials. So what I hope I've shown you is that starting out an academic career with a PhD, going into research, you can actually take your fundamental basic science right the way to clinical utility, and what I hope in this pandemic will have widespread utility. So let's now spend a few minutes before I answer questions. I see that there are questions that have been popping up on, I've told you my career in the context of somebody who's stayed absolutely fixed in academic research. But as this slide shows you here, there are lots of opportunities, whether you come out with a BSc, a master's, or a PhD, whether you go on to complete a postdoc. Yes, there's academic research, but there are many different opportunities for those who've been educated with a scientific background. And indeed, this is another way of presenting it. The opportunities are absolutely widespread. So for example, if you choose, you absolutely don't want to stay at the bench or doing any sort of research anymore, with a scientific background, the opportunity to become involved in patent law, to be involved in filing a patent examination, patent protection. With your scientific background, you could understand how to protect the intellectual property surrounding a scientific discovery. So you don't even have to be at the bench, but you could use your expertise to do that. Yes, there are opportunities also to remain at the bench, but perhaps not lead a research program. You could become a hospital technician. You could become a research technician in a research laboratory. You could even join pharma or biotech and be involved in technical aspects as a technician in a wet lab research. So you don't have to be in academic research, you don't have to lead a program, but you can still participate in research. Another area which I think many people overlook is the opportunity with a scientific background to become a consultant. Now, certainly as somebody with my years of experience, I can bring a lot to bear on consulting, but that doesn't mean that with a scientific background, even with a bachelor's degree, and I say even, like, I don't mean to diminish it, that you cannot bring to the table a scientific knowledge that might allow a new startup you to consult, to offer advice, to suggest what other experiments might need to be done, what other areas you might need to approach before that biotech or that startup proceeds. You could help with what we call angel investors. So with with a PhD, certainly your scientific background will allow you to offer your expertise to those who perhaps have no background in science, but would like to invest in some sort of scientific technology. And you could do the same either to what we call angel investors or in the context of pharma and biotech. This slide, what does it tell you essentially? That careers in bioscience are quite widespread and I would argue limitless. So for instance, we've talked about research, whether it's in universities, research institutes, or even as a hospital scientist. So certainly I'm affiliated with the University of Toronto, but my laboratory is at one of the hospital research institutes. If you choose not to do research, there's no reason why you can't use your scientific background to become a teacher, whether a lecturer at a college or a university, or indeed a teacher within a school, post-secondary or not post-secondary, primary or public school or private school. So you can teach science, and if you have a love of science, why not go that route? There are those, again, who may not wish to be involved in research at the bench, but would like to become involved in what we call regulatory affairs. So before any drug is approved for clinical use, it has to meet a number of criteria. And if you were to work in regulatory affairs, you would prepare the dossier, whether it's for a pharmaceutical company, whether for a biotech company, or any sort of startup, you would prepare the dossier to make sure that all the criteria are met for that drug to be approved. Let's say you don't want any of those careers, but you're interested in sales, to promote medicines, to promote equipment or technologies. Medical sales absolutely is looking for people with a scientific background who can communicate the science for sales. You could become what we call a science administrator. So there are many institutes, research environments, whether academic, within universities, colleges, or research institutes, that require administrators who have a scientific background to help coordinate those research engines. Again, within a company that's producing a medical device or a drug or a vaccine or whatever, production, quality control, it's important that people involved in the production or the manufacture of these drugs have that scientific background. So you can do all of that, or you could indeed be a communicator, whether that's in journalism, whether that's with a scientific journal, whether it's with a newspaper or any sort of media outlet. You could become involved in communicating research. And there's a real need, because many scientists are really very poor communicators. So to communicate science to the public would be important. In the lower panel here, I show you that scientific research requires many different areas to translate to the public sector. And there are opportunities, again, for somebody with a good scientific background to get involved. And the important point is, if you come out with a PhD, you have that rigor and you have that training that will allow you to get into so many different areas. In biotech, I've gone through all the opportunities that are there, whether it's at the bench, whether it's as an administrator, whether it's in regulatory affairs, whether it's helping with patents, lots of opportunities. And another important area is, as I said, engagement with the public sector, science communication, whether that be in journalism, whether that be to the government, even in areas like in museums where they have scientific exhibits. They need people to be able to communicate effectively. Then there's that whole area which is burgeoning now, which is bioinformatics and artificial intelligence. Essentially what that means is you take all the biological data that folks are generating, whether as research scientists, in whatever venue they're working, and you retrieve that data, you analyze it, you organize it, and you present it in such a way that the data, and now we're using these huge platforms with millions of data points. It's impossible just to look at the data on an Excel spreadsheet and make sense of it. If you have somebody who has skills in bioinformatics, they're really in demand now, how they can take that data, mine it, and help the scientist. So lots and lots of careers, I hope I've demonstrated to you. And perhaps the last one, which many of us are becoming more and more involved in, is helping shape policy, whether it's at the government level, whether it's in research institutes, engaging in debates with the public. Just as I said, was communication directly through the written word in journalism and various media. It's really important as scientists that we engage with scientific policy decisions. So what I hope I've done is demonstrated to you that a career in science can go from somebody like myself, basic science, all the way to having an impact, hopefully in the clinic and in pandemic scenarios or outbreaks, wherever. You can take your fundamental research and translate it if you choose to. And it doesn't have to be a medication, it could be a device just as easily. But if you choose not to take your scientific training, there are many, many opportunities that you could pursue. And with that, I think I'd like to thank you and open up to questions. So I will stop sharing my screen.
Dr. Fish, thank you so much. That was an incredible presentation, not surprisingly. We have 3 or 4 questions. Actually, we have 5 questions. Let's start with Colette's. Colette was wondering whether viruses could develop resistance to IFN treatment.
That's a really good point. So the answer to that is probably not. So when you make a pathogen-specific drug, and there are many now that are being developed, which are very specifically targeted to this SARS coronavirus spike 2 protein. Viruses mutate. This virus has already mutated from when it started in Wuhan to where it is in, for example, in Toronto. So viruses mutate, and they will mutate to become drug resistant. When you have something like interferon, I told you that it has multiple targets. It can hit the virus at multiple stages of its virus replication cycle, plus it can boost an immune response. So even if the virus were to mutate to block 1 or 2 or 3 or 5 different interferon targets, it's highly unlikely that it will ever mutate to the level where it would completely block an interferon response. So my answer to you is no, I don't think we will develop resistance to interferon.
So Dr. Fish, just to piggyback on Colette's question, can you talk about mortality rates? It seems to me that mortality rates appear to be going up based on the number of cases that are reported through secondary sources. So that may be the devil in the details here, but maybe you can comment on that.
So I think there are 2 things to be aware of. If you look to a country like Iceland, it's a small population. They have tested every single individual in their population. So they can then give an absolute percentage of those who developed coronavirus and those who died as a consequence. Unfortunately, in many jurisdictions, and Canada is one of them, first of all, across the country and across our provinces in Canada, testing rate is different. So you never know the number of confirmed cases. What you can confirm is the number of deaths. So we can absolutely say we have X number of deaths as a consequence of COVID-19, but what percentage that is of the total population remains unclear. What we do know is that there are those who are extremely vulnerable, seniors, and we've seen in long-term care facilities how it's decimated and killed many, and also those with what we have comorbidities. So if you have hypertension, if you have diabetes, the number of diseases, you're at higher risk of a more severe infection. So until this pandemic is over and done with, and we've collected all the data, and it really has to be rigorous, we cannot say absolutely what's going on in terms of fatality, but the indications are that it is less fatal than SARS coronavirus.
Thank you.
2002.
So let's move to, we've got quite a number of questions here. Pamela asks how she can get into the field of antimicrobial research. Any insight into that?
So Pamela, I don't know what your level of education is. If I'm going to assume that you're a university student. So let's say you are doing a Bachelor's of Science at a university. You would then go on to, first of all, see whether you're interested in research. So maybe during your undergraduate degree, volunteer or try and do some, get a course where you can do some research in somebody's laboratory, either over the summer or during the academic year. And if you like it, then the next stage is to say, okay, I need to get a higher degree, and that would be a master's or a PhD, which takes a considerable amount of time. A PhD is a commitment of, certainly in Canada, 5 to 6 years. And you'd find a lab that is involved in an area where you have an interest. So if you have an interest in microbial research, you'd need to go look at the various departments, immunology, microbiology, look at what the various professors, their research is, and see if they take you on as a graduate student. After your PhD, what you then have to do is do what we call postdoctoral fellowship. Again, this could be in an academic institution, or it could be at a pharmaceutical company or a biotech company. So there's a progression which you have to follow. You have to get the calling cards, but provided you have the appropriate academic training and you've got good hands in the laboratory, there's no reason why you can't get into this kind of research or activity.
Thank you, Dr. Fish. We have some great questions here. Eliza asks, even though interferon accelerates healing, does it necessarily prevent people from dying of the disease?
So that's an excellent question. So what we've learned is that it's not the virus that kills you. If you're going to die from SARS coronavirus, what the virus does is it sets up a sequence of events that lead to this incredible inflammation in the lungs. And it's this inflammation in the lungs, this inability to respire effectively, that puts people on ventilators. And associated with that, sometimes they have organ failures. So there's a cascade of events. So the notion is that you treat early while there's a lot of virus around to prevent going on to the next stage. So I would argue, and this is what the trials that we're looking at now, is to treat those individuals who are very early on in their virus infection so they never get to that severe stage.
Thank you, Dr. Fish. So we'll take one more question. Amy asks, are there any dangers to interferon treatment or any serious side effects noted? Do you want to speak to that?
Sure. So with chronic virus infections like hepatitis C, we've learned that when people are on interferon treatment for years, they do have side effects, but they're all manageable side effects. What we're talking about here is an acute virus infection, whether it was SARS, whether it was Ebola, where we treated with interferon, or now with COVID-19. In no patient did we see any exacerbation, any worsening of their disease. In fact, they improved. We didn't see any adverse events. So there's a fear that interferon will create what's called a cytokine storm. We didn't see that. We've done all the measurements. So my answer to you is no, I don't think, with the small numbers that we've looked at so far, and they are small numbers, that we definitely didn't see any adverse events. We didn't have to dose withdraw or dose reduce. So no, there were no adverse events that we could see at all, and the patients all recovered. But until we do a much larger randomized controlled trial, where we have many more numbers, we might see with many more numbers that there might be some adverse events that pop up. And again, it's always a balancing act. You know, if somebody has severe disease, are they prepared to take some uncomfortable adverse events to eliminate the likelihood of death? You know, that's what we're talking about. But I'm fairly confident, certainly from our preliminary data, than a number of different virus infections that we've treated humans, we haven't seen adverse events.
Thank you. Just to piggyback to your excellent comments, Dr. Fish, and it may be a rhetorical question, are we doing enough testing?
Well, you know, that's a good question. So I would say I am disappointed that we're not testing more widely. And why would you want to test? Because you want to test to see where there are clusters of infection. If you know where those clusters are, then you can self-isolate, or you can go in with a treatment when we have a treatment. If you don't test, you don't know where the clusters are, you don't know where there are community outbreaks. So it's to keep self-isolating people becomes problematic. You know, if nobody ever steps out of their house ever again, we will get rid of COVID, for sure. But it's not sustainable. So the best case is to test. And whenever there's a positive, you clamp down. Right now we isolate, but if you have a treatment, that's even better. Then you eliminate the virus. So absolutely, testing is really important. And not only testing, but what we call contact tracing. Because if you find a positive person, you need to know who that positive individual has interacted with that may also become infected, because they need right now to self-isolate as well. So absolutely testing and absolutely contact tracing.
Dr. Fish, how difficult is contact tracing, practically speaking?
It requires either an incredible number, a decent number of individuals who do this physically on the phone, tracking down people, or there are a number of technologies that are being developed, which unfortunately people are very reticent about because of privacy issues. Perhaps the most exciting one, I think, might be the Google and Mac coming together, Apple and Google coming together to form an app, to develop an app that you would have on your Android, and essentially it's on all the time. So it will trace where you are. Anybody else who has the same app, it pings. Now, ordinarily, you don't know, they don't know, it's completely anonymous. If you become positive, then it is possible to activate that app to be able to contact all those people that were in 6 feet of you while you had the virus or while you were asymptomatic for a number of days. I personally, when that app's out there, I'm going to get it. I think it's great, and because I own the ability to activate it or not. So my privacy is in my hands.
Wow, it sounds like there are a whole bunch of careers waiting to happen out there. I mean, you've really outlined that well for us. I'm going to wrap up. I want to thank you, Dr. Fish, for an incredibly informative session, both from a careers perspective and as a matter of obviously the utmost importance to humanity. What could possibly be more important right now to everybody experiencing, hopefully not the new normal. I also want to thank the University of Toronto's New College and Trinity College for their support in getting the word out to students during what could only be, a very difficult time. And in particular, Brenda and Catherine in alumni relations, they have been very supportive of my own initiatives over the years and in relation to this one. Everybody, please follow us at Career Path Intelligence on LinkedIn, and we'll have a new site up tomorrow. I apologize for the technical glitches that we experienced early on. We'll have to edit that out. The first couple of minutes when we were live, that falls on us. But you will have an opportunity to respond to a survey about our inaugural session, and we appreciate the feedback. Finally, don't forget to join us next week, May 11th, at 2 p.m. to hear immigration lawyer Catherine Sawicki talk about how COVID-19 has affected the status of international students and postgraduate work permits. This and every other town hall that we host will be free. So thank you, everybody, for supporting us. Thank you, Dr. Fish.